Hello everyone, and welcome to my presentation today on biochemistry, the fascinating study of molecules and chemical reactions within living organisms. What is biochemistry? Biochemistry is the scientific discipline that investigates the various molecules and chemical reactions observed in living cells and organisms. It dives into the intricate mechanisms that govern life processes at the molecular level. The primary goal of biochemistry is to unravel the mysteries of life by deciphering the language of molecules. Through the application of chemistry's concepts and techniques, biochemists aim to understand the molecular structures and functions that underlie biological phenomena. Life as we know it is primarily dependent on a select few elements: carbon, nitrogen, oxygen, hydrogen, sulfur, and phosphorus. These elements form the building blocks of biological molecules and are essential for the functioning of living organisms. On this slide, we will be talking about RNA. RNA stands for ribonucleic acid, and it is more versatile than DNA. Unlike DNA, RNA is capable of catalyzing chemical reactions, making it essential for various biological processes. Its ability to act as a mediator in protein synthesis and gene regulation underscores its significance in cellular functions. All living organisms are composed of one or more cells. Despite differences in complexity, cells across different organisms share similarities in size and basic structure. The classification of organisms into eubacteria, archaea, and eukaryotes reflects distinct cellular architectures and evolutionary pathways. Many essential biological substances consist of giant molecules that are polymers of simple monomer units. These biopolymers include polysaccharides, proteins, nucleic acids, and lipids, each playing crucial roles in cellular functions and metabolism. The diverse functions of these giant molecules highlight the complexity of biological systems. On this slide, we will be discussing Coulomb's law, water, and the Henderson-Hasselbalch equation. First, we have Coulomb's law, which shows our understanding of how charged particles interact within the cellular environment. It helps explain the attraction and repulsion between charged molecules, ions, and fragments within the cell. By understanding Coulomb's law, we gain insights into the electrostatic forces that govern molecular interactions, which are crucial for cellular functions. Next, we have water. Water is crucial to sustain life. Its significance in biology cannot be overstated, as it is the foundation of life as we know it. Its polarity and hydrogen bonding contribute to its ability to dissolve various substances, facilitating biochemical reactions within the cells. Understanding water as a solvent helps explain its role in transporting nutrients, facilitating chemical reactions, and maintaining cellular homeostasis. Lastly is the Henderson-Hasselbalch equation. This equation is a fundamental tool in understanding acid-base equilibria in biological systems. It allows us to predict how changes in pH affect the distribution of weak acids and their conjugate bases. By applying the Henderson-Hasselbalch equation, scientists can analyze and manipulate biochemical reactions dependent on pH, such as enzyme activity and protein structure. If you look at the formula here, that shows Coulomb's law. Alright, so in this slide we will talk about macroions. Macroions exhibit fascinating properties influenced by pH levels and the presence of small ions within their environment. First, let's explore amphilites. An amphilyte is characterized by possessing both acidic and basic ionizing groups within its structure. Depending on the pH of the solution, an amphilyte can exhibit a net positive, neutral, or net negative charge, which directly impacts its behavior and interactions within the solution matrix. Moving forward, we have polyamphilates. These stand out for their complexity, featuring numerous acidic and basic groups distributed throughout their molecular structure. This abundance of ionizing groups lends polyamphilates diverse properties and behaviors, contributing to their unique characteristics in solution. Finally, we have polyelectrolytes, which are compounds distinguished by multiple ionizing groups carrying a singular charge. The behavior of polyelectrolytes is heavily influenced by the ionic strength of the solution, with the magnitude of screening determined by the presence of small ions. Understanding these interactions is crucial for comprehending the behavior and dynamics of macroions in solution environments. Next up, we will talk about the fundamental thermodynamic principles that explain how organisms harness energy from their surroundings to power essential cellular processes. Thermodynamics provides a framework for understanding the directional flow of energy and reactions within biological systems. Central to this understanding is the thermodynamically favored direction of chemical reactions, which is dictated by changes in both enthalpy and entropy. Enthalpy represents the heat content of a system, while entropy reflects the system's degree of disorder. The concept of free energy, expressed as G equals H minus Ts, encompasses both enthalpy, or H, and entropy, or S, and T, which is the temperature in Kelvin. This equation allows us to predict whether a reaction will proceed spontaneously or require energy input. Understanding these thermodynamic principles is critical for comprehending how organisms extract and utilize energy to sustain cellular functions and life processes. Here we will talk about energy changes within chemical reactions. First, let's explore exergonic reactions. These reactions occur when there's a negative change in free energy. In simpler terms, they release energy as they proceed. This energy release is often harnessed by living organisms for various biological processes. Conversely, endergonic reactions are the opposite. Here we observe a positive change in free energy. In other words, energy is absorbed rather than released during this reaction. These reactions typically require an input of energy to proceed, and they often drive vital cellular processes. It's worth noting that not all reactions proceed spontaneously. Even if a reaction isn't thermodynamically favored, it can still move forward if coupled with reactions or processes that have significant changes in free energy. This coupling mechanism is crucial for sustaining life processes and maintaining cellular equilibrium. Moreover, concentration gradients across membranes serve as important free energy stores within cells. These gradients represent potential energy that can be utilized for various cellular functions, including active transport and ATP synthesis. Understanding and harnessing these energy gradients are fundamental to cellular metabolism and function. The structural organization of proteins. The primary structure of a protein refers to the fundamental and initial level of organization within its structure. It represents the linear sequence of amino acids that constitute the backbone of the protein molecule. Each protein possesses a unique primary structure determined by the specific sequence of amino acids it contains. This sequence is encoded in the genetic material of the organism, particularly within the DNA. The primary structure lays the foundation for the higher order structures and functions that proteins can exhibit. It influences the protein's folding pattern and its ultimate 3D shape, which in turn determines its biological activity and functionality. Next, we have secondary structures. Secondary structures are the organized spatial arrangements of neighboring amino acid residues within a polypeptide chain. It's interesting to note that there are three primary types of secondary structures: the alpha helix, the beta sheet, and the beta turn. The alpha helix forms a spiral structure, while the beta sheet consists of parallel or anti-parallel strands. The beta turn, on the other hand, creates a sharp turn in the protein chain. Moving on to the tertiary structure, we encounter the complete 3D shape of a protein molecule. This structure is critical as it defines the specific conformation and functionality of the protein. Now we will discuss polymers, specifically focusing on proteins and amino acids. At the core of biological processes, proteins stand as polymers of L-alpha amino acids. These complex molecules are pivotal for the structure, function, and regulation of cells within living organisms. Oligopeptides and polypeptides are fundamental components in protein synthesis. These chains emerge through the polymerization of amino acids, facilitated by peptide bond formation. Notably, the peptide bond, which is a crucial link in this process, adopts a planar structure, with the trans form being the favored conformation. Within the realm of genetics, the genetic code orchestrates the synthesis of proteins by encoding for 20 common amino acids along with 2 rare amino acids. These amino acids are assembled into polypeptide chains during the process of translation, where the messenger RNA, or mRNA, guides the synthesis of proteins at the ribosomes. On this slide, we'll talk about proteins. Proteins represent fundamental molecules within the cell, synthesized through the energy-dependent process of translation, driven by ATP. This complex process follows the genetic code, composed of codons, which read sets of 3 nucleotides, each specifying a unique amino acid. With the arrival of genome sequencing, an extensive repository of gene sequence information has become available, which illuminates the molecular complexity of protein synthesis. Notably, proteins exhibit polyamphylitic properties, as their side chains possess both positive and negative charges. Understanding these molecular characteristics shows the complexity and versatility of proteins within cellular functions and biochemical processes. Proteins exhibit a remarkable diversity in their structures, which directly influences their functions. The sequence of amino acids in a protein, which is encoded by genes, serves as the blueprint determining its overall structure and function. It's essential to recognize that protein sequence plays a pivotal role in dictating various levels of protein structure, including local folding, also known as secondary structure, global folding, also known as tertiary structure, and organization into multi-subunit structures. At the secondary structure level, proteins fold into recognizable patterns such as alpha helices and beta sheets. These structural patterns emerge from specific patterns of hydrogen bonding between amino acid residues along the polypeptide chain. These secondary structures are the building blocks that contribute to the overall shape and function of the protein. The complexity of protein folding and structure is aided by tools like Ramachandran plots. These plots provide a visual representation of the allowed conformations of amino acid residues in a protein by plotting the phi and psi angles of the polypeptide backbone. They offer insights into the structural arrangements and constraints that govern the formation of secondary structures within proteins. Lastly, we will be talking about proteins and how they are grouped into 2 broad categories. The first category is the fibrous proteins. These proteins are characterized by their elongated shapes and regular secondary structures. They primarily serve structural roles within cells and organisms, and they provide stability and support to cellular components and tissues. In contrast, we have globular proteins, which possess more complex tertiary structures, folding into compact shapes. These proteins exhibit a diverse range of functions, including enzymatic activity, molecular recognition, and transport. Various factors contribute to the stability of globular proteins, including hydrogen bonding, hydrophobic interactions, and disulfide bonds. The folding of globular proteins typically occurs spontaneously and rapidly under native conditions, reflecting the intricate and finely tuned mechanisms underlying protein structure and function within biological systems. Understanding the distinction between fibrous and globular proteins is crucial for unraveling the complexities of cellular processes and organism functions.